All right, so the best way to understand anything in genetics or molecular biology, or probably anything at all, is to try to start applying that information. So I don't usually have you do a whole lot of listing of things, but these are the questions and problems that I thought illustrated the most important parts about DNA. So, what four general characteristics must the genetic material possess? It needs to be complex, right? Because we need to get a lot of information out of it, and uh, so complex. It has to be able to be complex because there's complex information. So that means there has to be a complex structure. And I guess I can talk a little bit more about that. It has to be able to be replicated, of course, because we know that the definition of the genetic material is what molecules are carrying the information that's passed down from cell to cell, and from uh, mom to I was going to say mom to dad, but mom to kid, mom and dad to kid. So you have to replicate that information if you're going to pass it down. You have to be able to express that. There has to be some mechanism for expression, and then it has to be able to be variable. So those are the four things. All right, so I'm going to expand on that a little bit because there's some interesting. I think we talked about the structure of DNA, but I'm not sure we talked about exactly how that relates to all this stuff. So let's do that now. So the complex nature of it, the molecule, I guess, isn't the individual nucleotides aren't all that complex, but when you put a whole bunch of them together, once Watson and Crick figured out that the DNA was a big long sequence of information, they came up with the idea that there must be some kind of code. Because you have four different nucleotides and you can make it as long as you want. The human genome's 3 billion of these things linked together. You can come up with lots of different combinations of things. So just because you can have the four bases in any combination gives you the ability to encode complex information. Then the other thing, this double-stranded nature of DNA, and more importantly, this rule that G's always have to pair with C's and A's always have to pair with T's. That means that you can separate the two strands, and then you immediately know what to put on the opposite strand. Now we're going to talk about DNA replication in a decent amount of detail, but this is the basics: is that you can separate the two DNA strands, and when you separate them, you you copy the one strand and you put in the complementary base, and by doing that, you end up with two strands that are identical to what you started with. So if we start out with AGC, we'll just do a short one, AGCGA and TCGCT. We separate those, and we got AGCGA here, and then TCGCT here, and then we copy it, and we copy this one. It's the exact same molecule. So that's the exact same as this, and that's the exact same as this. So we've copied it, we've replicated it. Expression, we need to go from DNA to RNA. We'll talk about that in more detail later, and we'll talk about mutations later as well. But the most important thing that they came up with, they didn't really come up with the expression part or the variable part for a long time. But the first part was just that it had to be complex enough to store information, and that there had to be some obvious mechanism of replication. And Watson and Crick, in their first paper, when they described the structure, or maybe it was the second paper, they came up with that idea and said this They said something like, it doesn't escape our attention that this structure of DNA leads to a mechanism of potential replication. So, draw and identify the three parts of a nucleotide. Draw a house with a chimney. That's your sugar. Throw on a base. Just give it an A. Give it a phosphate. So that's like the abstract version. Draw it out a little bit more for a DNA nucleotide. That has to be an H, an OH. Probably has 3 phosphates here. You don't really usually draw out all of the oxygens there, but we could. And it's got the base. And you have to know that's 1 prime, 2 prime, 3 prime, 4 prime, which is in there. There's another carbon there. That's 5 prime. All right, simple enough. RNA differ from DNA. RNA has a U. DNA has a T. RNA is single-stranded. RNA has ribose. DNA is double-stranded. DNA has deoxyribose. But most importantly, RNA forms complex secondary structure. So it forms all these loops and whatnot. And the whatnot's pretty important. Your loops are important, but the whatnot is really important. And you know, it doesn't look anything like that. tRNA is the easiest one to conceptualize. So there's these, because RNA is single-stranded, it can form these different complex molecules. How is a purine different from a pyrimidine? Well, purine has a double ring structure. You know, one of the rings has 6 carbons and the other one's a 5 carbon ring. So if you know that, there's random miscellaneous double bonds in there. We got some nitrogens in there somewhere. Pyrimidines are single-stranded structures. And the purines in DNA, so if we go, let's say, we'll go DNA and RNA, we'll have our purines and our pyrimidines. In DNA, it's A and G. In RNA, it's A and G, so the purines are always A and G. The pyrimidines are C and T in DNA and C and U in RNA. Draw a short segment of a single polynucleotide strand, including at least 3 nucleotides, indicate the polarity. All right, so 3. Okay, so that's a pain in the neck. Let's see if I can make some room over here. So this will keep our phosphates in there now, just so we can show this phosphodiester bond. Important that these are negative. Oh, not the double bonded one isn't negative. So there's 2, and then the third one is here. And let's say that's an A and a G and a T, whatever it is. This is our 5 prime carbon. It's got a phosphate on it. So that when they say polarity, that means direction. So what direction does it go in? 5 prime to 3 prime. And this 3 prime is going to have a 3 prime OH. And it didn't say here, it didn't say whether it was DNA or RNA, but we'll just make it DNA. And so there's just a hydrogen there. What bases are capable of forming hydrogen bonds? A and T can form 2. G and C can form 3. And this other one kind of makes you think about the different kinds of bonds. So let's say we've got a DNA molecule. Oh, I'm going to get an A in there. That's not good. TCG, maybe 5 prime, 3 prime. So we got hydrogen bonds. And then we've got These guys are phosphodiester bonds. I think that's about it. And the key is that these are covalent. And H bonds are H bonds. So what are some of the important genetic implications of DNA structure? Well, the fact that, like we talked about before, that it can be replicated. The GC rule and the AT rule tells us that it can be replicated. The fact that it's double-stranded means it's stable and it doesn't form into weird different structures and do that sort of thing. So it has to be stable if it's going to be, store all the genetic information and pass it down to generation to generation. So basically, it's that the genetic implications are that it can do those four things that we showed up at the top. So it can be easily separated. So the fact that it's hydrogen bonds means that it can easily be separated into two strands, and then you can, because A's pair with G's, you know what sequences to put when you make new DNA. You know what sequence to make the other strand. So the genetic implications in my book are mainly the A and T base pair rule and the GC base pair rule allow you to copy it. What are the three major pathways of information flow? Three? Oh yeah. So this is the central dogma of molecular biology. Um, so um, three major pathways of information flow. All right. So this is kind of the theme of this whole section. So DNA, we just said up there, can be replicated. So we draw that like this. DNA can kind of make itself. So replication. Transcription. And translation. You really have to have this straight. If you're writing an answer to a question ever and you say transcription or translation, or transcription instead of translation, you'll probably get a point off every time you do that. So it's important to make sure you know what transcription is, you know what translation is, what kinds of things happen during transcription, what kinds of things happen during translation, and DNA replication as well. So that's the flow. Call this the central dogma. And the idea is that it's all about information. It's information flow. What are hairpins and how do they form? They're things that look like this, and they form by intramolecular base pairing, as opposed to intermolecular base pairing. So that's two separate DNA molecules that are held together by base pairs. This is intramolecular because it's within the same molecule. DNA molecules of different sizes are often separated with the use of a technique called electrophoresis. You've all done electrophoresis and run gels. With this technique, DNA molecules are placed in a gel. Electric current is applied. So let's say we're going to put the DNA here. We would normally make this vertical. I should probably do it like this. So you almost always draw gels like this. You put the DNA in here. And so let's say this is our DNA. And then we turn on the juice and the DNA runs in this direction. Well, it'll run in that direction if we have a negative pole here and a positive pole here. And DNA runs away from the negative and towards positive. Why is that? Because those phosphates are negative. Those negative charges on the phosphates. So here's our bases hanging off. Here's our phosphodiester bonds between here. Got our sugar, phosphates, and the phosphates are negatively charged. And so if you have a big, huge, long molecule of DNA, it's going to have tons and tons of negative charges. one nucleotide strand as this, right in the base sequence, and label the 5' and 3' ends of the other strand. This is, most people usually get this right, but I've seen people get it wrong. It's really important that you have anti-parallel strands all the time. And that A pairs with T and T pairs with A. It's certainly easy. You've already seen me screw it up at least once. It's easy if you go quickly to screw it up, but you can indicate your hydrogen bonds like that and put 3 of them between here. Didn't really ask for that. But you really just need to know the term complementary and be able to, polarity and anti-parallel and all that kind of stuff. So if a double-stranded DNA molecule is 15% thymine, well, so that's your T, then it's also 15% adenine because they always have to match up, especially if it's, only if it's double-stranded. So that, if you add that up, that's 30, and 100 minus 30 equals 70, and half of 70 is 35, so the other guys have to be 35. So this would maybe be G's and C's. So if you know the percentage of one, you can figure out the percentage of the other, and then once you know that, you can figure out the percentage of the rest. And this is our last one. Well, almost our last one. The relative amounts of each nucleotide base are shown here. And this is a virus. Now, viruses can have anything. They can have DNA, they can have RNA, they can have single-stranded DNA, single-stranded RNA, double-stranded, whatever. So in this one, we can look at these things and we can decide if it's DNA or RNA and if it's single-stranded or double-stranded. So virus 1 has U. Anything that has U's in it and not T's, that's RNA. Because C and G are equal and U and A are equal, it's double-stranded. This one has T's, so it's DNA. T and A are equal, G and C are equal, so it's double-stranded. This one has DNA because it has T's. And T and A, those are pretty close, but the C's, there's way more C's, so that's single-stranded. This is RNA because it has U's. C's and G's are pretty close, but U's and A's are far apart, so that's RNA, and it's also single-stranded. So it's double-stranded if it's double-stranded, then your A's and T's have to match up, and your T's and A's have to match up, and your G's and C's have to match up. But if it's single-stranded, you can have any number of these things, and G's and C's and A's and T's won't have any relationship with each other. All right, just one last thing. Write out the basis of an RNA molecule that will form a hairpin structure. So a hairpin structure is something that looks like this, and all we have to draw is something where there's some kind of base pairing rules that will allow these things to fold up. So the simplest way, whenever I do it, just to kind of keep it straight, if you just put all A's down here, actually it's asking for just a hairpin, not a stem loop. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. So there's a sequence that could easily, we'll just, there's only 4 of them, so those can easily form together into a secondary structure or a hairpin. If we had something like this, a bunch of A's, and then maybe a G and A here, and then a bunch of U's, then we could, all the A's could come together, the G and the A in the middle could loop out, and then those A's could go like that. So it's just the idea of showing that RNA molecules can form hairpin structures by what we called intramolecular H bonding. All right, so that's it. So we'll stop that one there. That was only 19 minutes.